Hello friends, welcome in another video lecture of Easy Life Science. Here I am talking about transcription in prokaryotes. So please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon so you will get the notification of my newly uploaded video. So let's start. The central dogma of a life. The central dogma of life says that the DNA makes its copy through a process that is a replication, and the genetic information stored in a DNA get converted into a messenger RNA, and this process is known as a transcription process. Further, the genetic information stored in a mRNA in form of codon, it converted into a amino acid to formation of a protein, and this process known as a translation process. Sometimes the RNA get reversed into a DNA, and this is known as a reverse transcription. So this is a central dogma of life, and this concept was given by a scientist Watson and Crick. The transcription is a synthesis of a RNA from a DNA, or it is a synthesis of a mRNA from DNA. But in case of a reverse transcription, there is a synthesis of a DNA from a RNA. For example, in retroviruses, and this concept of a reverse transcription was given by the Themin and Baltimore in a Ross sarcoma virus. And this phenomenon is known as theminism, and the enzyme catalyzes this reaction is a reverse transcripted enzyme, or it is also called as a RNA dependent DNA polymerase enzyme. Once a mRNA is formed or a messenger RNA is formed, the message in form of a codon, it is converted through a synthesis of a chain of amino acid to formation of a protein, and this is known as a translation process. The transcription means a synthesis of a RNA from a DNA. The genetic information stored in a DNA is converted into a messenger RNA, and this is known as a transcription process. Three types of RNA are there: messenger RNA, ribosomal RNA, and a transfer RNA. These are synthesized by a transcription of appropriate gene and involved in a protein synthesis. The messenger RNA carries the message from DNA to the ribosome. The ribosomal RNA, it is a major structural component of a protein synthesizing ribosome, and the tRNA or a transfer RNA, it acts as a adapter molecule in aligning the amino acid according to a sequence present in a mRNA. Now the transcription in a prokaryotes. Transcription in prokaryotes and transcription in eukaryotes are different. Transcription is catalyzed by an enzyme RNA polymerase, which makes a RNA using a DNA as a template. The strand of a DNA which is transcribed is a template or a sense or a non-coding strand, and this strand runs in a 3' to 5' direction. The other strand, called as nonsense or non-template or a coding strand, which runs in a 5' to 3' direction, and this strand is identical to the strand or a RNA transcript. The prokaryotic organism have only one type of RNA polymerase which synthesizes all the three types of RNAs. Whereas in case of eukaryotes, three types of RNA polymerases are there: RNA polymerase first, second, and a third. The transcription requires the double helix DNA should be partially unwound in the region of mRNA synthesis. The region of unwinding is called as a transcription bubble. Formed mRNA strand is complementary to the DNA template and is almost identical to the other strand, that is non-template or a nonsense or a coding strand. Only difference is that in mRNA all the thymine nucleotides are replaced with uracil nucleotides. In an RNA double helix, adenine can bind to a uracil via 2 hydrogen bonds, just as a adenine paired with the thymine in a double stranded DNA helix. The transcription process is catalyzed by an enzyme RNA polymerase. The enzyme RNA polymerase is a hollow enzyme and it is formed from a core enzyme and a sigma factor. The core enzyme made up of from 5 polypeptide chains alpha, alpha, beta, beta prime and omega. In association with a sigma factor, a complete enzyme is formed or a hollow enzyme is formed known as RNA polymerase. The prokaryotic RNA polymerase and eukaryotic RNA polymerase are different in their types of subunit and a number of subunits. RNA polymerase is an enzyme that synthesizes RNA from a DNA template using another enzyme, that is a helicase. RNA polymerase locally opens the double stranded DNA so that one strand of exposed nucleotide can be used as a template for a synthesis of RNA. A transcription factor and its associated transcription mediator complex must be attached to a DNA binding site called as a promoter region. RNA polymerase moves along the template strand of DNA in the 3' to 5' direction and the RNA transcript grows in a 5' to 3' direction. The 5 polypeptides in a core enzyme are alpha, alpha, beta, beta prime and omega. The alpha subunit having a molecular weight 36 kilodalton, the beta subunit having a molecular weight 150 kilodaltons, beta prime has a molecular weight 155 kilodalton, and this is a small omega subunit, and this forms a core enzyme. The sigma factor binds to a core enzyme at time of a transcription initiation, forming a hollow enzyme, that is a RNA polymerase. Hollow enzyme is a complete enzyme. Core enzyme plus a sigma factor forms a hollow enzyme or a complete enzyme. After transcription start, the sigma factor can get unwind or it get disassociated from a core enzyme and let the core enzyme proceed its further RNA synthesis. Stages involved in a transcription. The first stage, binding of a RNA polymerase to a template DNA strand. The nucleotide pair in a DNA double helix that correspond to the site from which the first 5' mRNA nucleotide is transcribed is called as a +1 site or a initiation site or a start point. Here in this image there is one DNA. In this DNA, the strand which runs in a 3' to 5' direction is a template strand, and this strand get converted into a messenger RNA. This strand is a non-template strand and Here is a start point or a +1 point from which the first 5' nucleotide of a RNA is formed, that is a adenine. Nucleotides preceding the initiation site are given negative numbers and they are designated as upstream sequences. Conversely, the nucleotide following the initiation site are designated as downstream sequences, or they are given as a positive numberings. It means that the nucleotide which are present preceding to this start point are given negative numbers, and these are called as upstream sequences, whereas the nucleotide which are following this initiation site are given positive numbers and called as downstream sequences. Prokaryotes do not have a membrane enclosed nuclei, therefore the process of transcription, translation and mRNA degradation can all occur simultaneously. The intracellular level of a bacterial protein can quickly amplified by a multiple transcription and translation events occurring concurrently on the same DNA template. Prokaryotic transcription often converts more than one gene and produces a polycystonic mRNAs that specify more than one protein. On a DNA, there is one region called as a promoter. A promoter is a DNA sequence onto which transcription machinery binds and initiates the transcription. In most cases, promoter lies upstream of genes that they regulates. Although promoters vary among prokaryotic genomes, a few elements are conserved. The DNA promoter region is a stretch of about 40 base pairs and including a transcription start point. A promoter have a start point designated as a +1 and usually it is a adenine. At upstream or at a -10 and a -35 region, here we can see that a -10 sequence or a -35 sequence, it is a upstream of a start point, and as it is upstream it has given number as a -10 and -35. These sequences are consensus sequences. At the -10 and -35 regions upstream of initiation site, there are two promoter consensus sequences or a region that are similar across all promoters and across various bacterial species. The -10 consensus sequence is of 6 nucleotides, that is TATAAT. The -35 consensus sequence is of 6 nucleotides TTG and AGA. These two consensus sequences are recognized and bound by a sigma subunit of RNA polymerase. Initiation. The RNA synthesis start from a +1 nucleotide in a promoter sequence of a DNA. The RNA synthesis start from this point. But before this, the adenine and thymine rich -10 region facilitates, that means this region, adenine and thymine rich region, which is -10 or upstream to the start point, facilitate the unwinding of the DNA template. Sigma subunit of RNA polymerase recognizes these -10 sequences upstream of a transcription start point ribonucleotide triphosphates, that is a RNTPS, acts as a substrate and a hydrogen bonded to a complementary basis of a template strand of a start point. At a start point, RNA polymerase forms a phosphodiester bond between 3-OH end of a first RNTP and a 5' end of next RNTP, and two phosphates are released. After adding up to 9 RNTPs, the sigma subunit detached from a hollow enzyme, that is from a complete RNA polymerase, and the core enzyme carry its synthesis. Elongation begins with a release of a sigma from a hollow enzyme. The dissociation of a sigma allows the core enzyme to proceed along the DNA template, synthesizing a mRNA in a 5' to 3' direction at a rate of 40 nucleotides per second. As elongation proceeds, the DNA is continuously unwound ahead at the core enzyme and rewinds behind a core enzyme. The base pairing between template strand and a newly formed RNA transcript is not stable enough to maintain the stability of mRNA synthesis. Instead, the RNA polymerase acts as a stable linker between DNA template and a nascent RNA strand to ensure that the elongation not interrupted prematurely. It means this RNA polymerase is a linker between nascent RNA and a DNA template, so that the process of transcription does not get interrupted. The unwinding and rewinding by a RNA polymerase is relaxed by a topoisomerase. Here is a complete picture of a transcription. At a promoter, there is a binding of a RNA polymerase, and this binding of a RNA polymerase through a local unwinding forms a transcription bubble. The RNA polymerase reads a template strand from a 3' to 5' direction and makes their copies from 5' to 3' direction as like this, and a sigma unit from the complete enzyme get detached and the RNA polymerase runs in this direction, leads to a elongation of RNA. After elongation or a formation of a long RNA molecule, there are some termination signals present in a DNA template, so that the RNA polymerase falls down and stalls the transcription process, and from that newly formed transcript is released. Then the termination of a transcription. Once a gene is transcribed, depending on that gene, there are two kinds of termination signals. One is a protein based and other is a RNA based. The protein based termination is called as a row dependent termination, and other is called as a row independent termination. The first row dependent termination it is controlled by a row protein or a row factor. which tracks behind the RNA polymerase on a growing mRNA strand. Near the end of the gene, the polymerase encounters a run of G nucleotides. On a DNA template and it stalls. As a result, the Rho protein collides with the polymerase. The interaction with a Rho releases the mRNA from a transcription bubble. The Rho factor protein causes a disruption of DNA RNA hybrid. The Rho protein is specialized helicase enzyme which recognizes 50 to 90 base pair upstream of a terminator in a mRNA sequence. Rho protein releases a newly formed RNA. Here one schematic representation of a Rho dependent termination. When a RNA polymerase reach to a number of guanine nucleotides, the Rho factor collides with a RNA polymerase, and so that the RNA polymerase falls down, and hence from that newly formed RNA get discharged. Then next, the Rho independent termination. It is controlled by a specific sequences in a DNA strand. As a polymerase comes near the end of gene being transcribed, it encounters a region rich in a C-paired G nucleotide on a DNA double helix. The mRNA folds back on itself and the complementary cytosine guanine nucleotide bind together on a mRNA transcript and results in a stable hairpin structure. The hairpin causes a polymerase to stall as soon as it begins to transcribe a region on a DNA which is rich in adenine paired with the thymine nucleotides. The complementary uracil paired adenine region at the mRNA transcript forms only a weak interaction with the template DNA. This, coupled with a stalled polymerase, induces enough instability for a core enzyme to break away and liberate a new mRNA transcript. Here, this is a folding back of a RNA transcript where the guanine pairs with a cytosine and forms a hairpin like structure. When on a template DNA, the RNA polymerase finds out a reach of adenine nucleotides. It folds back and forms this hairpin like structure. It is interconnected by hydrogen bonds in between cytosine and guanine, and the RNA polymerase, because of this hairpin structure, stalls and stops its transcription, and this liberates a new mRNA transcript. So this is about the transcription in prokaryotes. Thank you for watching my video. Please subscribe my channel Easy Life Science. Like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So thank you once again.